Welcome back to Strive Sessions. I'm Rex, pharmacist with Strive Compounding Pharmacy. Today we're going to explore peptides. Be sure to stick around for your Q&A afterwards. Quick disclaimer before we start. This content describes general trends in peptide research and is intended for educational purposes only. References to clinical development, approved medications, and investigational agents refer to the broader scientific field and do not imply that any compounding preparation has been reviewed, approved, or investigated by the FDA. Compounded peptide preparations are not FDA approved and are distinct from investigational and improved peptide medications. All prescribing decisions should be based on a licensed healthcare provider's independent clinical judgment. So as we enter into a new era of personalized medicine, peptides are emerging as a very promising approach. They share structural similarities with the body's own endogenous signaling molecules and can address very specific physiological needs in patients through these natural signaling pathways. Peptides are shifting medicine towards a more signaling-based, patient-specific approach. We're going to cover 4 key areas about peptides today. We're going to get into peptide biology, where we'll talk about their structure and fundamental molecular properties. We'll talk about peptide signaling, where peptides interact with the receptors of the body to produce their biological effects. We'll get into peptide pharmacology, where we'll discuss key aspects of their pharmacokinetics and pharmacodynamics. And we'll get into clinical considerations, discussing delivery methods, pharmacological constraints inherent to peptides, and patient considerations relevant to peptide regimens and a 5-by-3 compounding setting. So why does understanding peptides matter in practice? Peptides are crucial biologically active signaling molecules that regulate various physiological processes. Your own endogenous peptides do this. And some key characteristics of peptide medications is that they share structural similarities with these natural signaling molecules and can interact with specific pathways in the body to support various physiological functions such as metabolism, endocrine signaling, cellular communication, and they can address very patient-specific physiological needs. It's important to know that FDA currently has many approved medications that are peptide-based across many medical specialties, and their peptide pharmacologic behavior can differ somewhat from traditional small molecule drugs. Their delivery method and formulation greatly influences their exposure and activity within the body. It's important to note that all prescribing decisions regarding peptide preparations should be guided by all available clinical evidence and appropriateness with all regulatory frameworks that are applicable. So what is a peptide? A peptide is a short chain of amino acids. These are the same amino acids that make up building blocks of proteins. And as I said, they typically act as biological signaling molecules in the body. Examples of your own endogenous peptides include insulin, glucagon, oxytocin, vasopressin, and many others. So what separates a peptide from a protein? This comes down to a matter of their size, their structure, and the typical biological roles. So peptides are typically considered to be about 2 to 50 amino acids long in their chain. Although this is not a hard biological line, it's more conventional. And as I mentioned, they function as signaling molecules. They commonly bind to receptors on the cell surface embedded in the cell membrane. And they are generally much smaller and less structurally complex than proteins. Proteins are often hundreds to even thousands of amino acids long. They have a wide variety of functional uses in the body. They might be structural components or enzymatic molecules. And they fall into very complex 3-dimensional structures. So let's go further into peptide structure to understand it better. These amino acid chains that peptides are made of, they're linear and they're held together by peptide bonds, and they have directionality. They go from the N-terminus to the C-terminus. The amino acid sequence that the peptides consist of is what determines their structure and function. Even a small change in the sequence can have a huge impact on its ability to bind with the receptor or have any biological effect. Let's discuss further how peptides act as biological signaling molecules. They can participate in many major roles in signaling in the body. They might participate in hormone signaling. They can modulate neurotransmitter activity. They can modulate immune responses, and they can facilitate growth and repair pathways. And they can participate in different modes of signaling. One might be endocrine signaling, where they enter the plasma and go off to interact with distant tissues and organ systems. They can participate in paracrine signaling, where they signal out to other nearby cells to coordinate a response. And they can participate in autocrine signaling, where the cell actually signals out back to itself to further a functional response or other identical cells. So in all, peptides coordinate communication between cells, between tissues, and between organ systems. So how do peptides produce these biological responses? I'm going to give a quick overview now of how this occurs. If you look at the visual, the peptide or the peptide ligand binds with a receptor normally located on the cell surface embedded in the membrane. When it binds with the receptor undergoes a conformational change or structural change. This structural change triggers an intracellular signaling cascade, and this amplifies the original activation signal from the peptide and causes functional responses in the cell. These functional responses then go on to change cellular behavior and potentially signal out to other cells to coordinate these responses. So let's talk more about the receptors that peptides interact with. I'm going to discuss three major families of receptors right now. The first is G protein coupled receptors. These are one of the most common receptors that are associated with peptide drugs, and they go on to activate what's called intracellular secondary messenger systems. Examples of these include GLP-1 receptor agonists, oxytocin receptor agonists, and GHRH receptor agonists. And then there's receptor tyrosine kinases. These activate intracellular phosphorylation cascades via autophosphorylation, and examples of this include insulin receptor agonists. And then there's cytokine receptor signaling family. These activate transcription signaling, often through the intracellular signaling cascade called the JAK-STAT pathway. And examples of this include growth hormone receptor agonists and erythropoietin receptor agonists. Now, these last two that I mentioned are both enzyme-linked receptors, and it's important to note that all these receptor families can have overlapping effects and crosstalk to each other, and actually not operate in isolation, but trigger intracellular cascades in all these receptor families. So let's go further into these intracellular signaling cascades to understand them. So the first that I brought up, the secondary messenger signaling cascades. This is when the body uses diffusible small molecules such as cyclic AMP or calcium to carry the signal inside the cell and amplify it. Examples of this, such as cyclic AMP signaling pathway might go on to support metabolic function and hormone signaling, and the calcium signaling pathway might go on to support cellular secretion, contraction, and neuronal activity. Then there's the phosphorylation cascades. This is when the body uses protein phosphorylation in an on-off switch type pattern to relay and amplify the signal inside the cell. And this might involve the MAP kinase pathway, which might be involved in cellular growth and gene expression, or the P13K AKT pathway, which might aid metabolic function, cell maintenance, and cellular growth. And then there's transcription signaling. This is when the signal is carried all the way down to the nucleus to affect gene expression. So this might actually kind of switch on and off how genes are operating, and they can go on to support hormone and cytokine signaling, cellular growth, and hematopoietic function. So it's important to know that there are some exceptions to how this works. Some peptides are actually capable of crossing the cellular membrane and triggering an intracellular cascade on their own without interacting with the receptor on the cell surface. Although this is a small number of peptides, they typically do this by interacting with what's called the AMPK intracellular signaling pathway. This direct intracellular activity might influence metabolism, the cell stress response, and also gene expression. The peptides that operate in this way are all currently still under investigation, but it is a large area of emerging research. So how do we get from these signaling cascades to an actual effect? I'm going to go through this in kind of like a time-wise type fashion. So the first thing that might happen is you might get modulation of protein activity. This can literally happen in seconds. And this might involve enzyme activation or inhibition, or further changes in signaling inside the cell. And these further changes in signaling inside the cell is where the overlap of these different pathways occurs that I mentioned. And then on the order of hours, you might get gene expression changes. This can be activation or suppression of transcription factors, kind of switching on and off genes, and then alter protein synthesis. And then these two mechanisms together converge to produce a cellular functional response. This might result in changes of cellular behavior, including changes in secretion, metabolism, growth, or contraction. And this is also when the cell signals out to other cells to integrate this response across cell populations. And this integrated response is what scales up into these larger physiological effects that you might see. It's also important to note here that the body tries to tightly regulate the signal intensity and duration of these responses so that in an attempt so that no abnormal pathology is to occur. So these now coordinated cellular changes scale into entire physiological responses that might be involved in metabolic outcomes, such as effects on glucose handling, energy balance. Think of insulin receptor agonists or GLP-1 receptor agonists and their effects. It might go on to affect endocrine signaling, where peptides modulate hormone release patterns or change how the body responds to hormone feedback in the endocrine axis. It might go on to modulate tissue adaptation, where growth factors and repair mechanisms might be affected, such as remodeling or extracellular matrix, or maintenance of cellular tissues might be facilitated. And then there might be immune modulation, where peptides might affect cytokine tone, leukocyte behavior, and inflammatory responses. So now let's get into the pharmacokinetics of peptides. Pharmacokinetics describes what the body does to a molecule. This includes how the molecule is absorbed, how it's distributed through tissues, how it's metabolized by enzymes, and how it's eliminated from the body. Some key pharmacokinetic characteristics of peptides includes that their molecular size often limits their ability to passively diffuse across cellular membranes, and this is why they are often interacting with receptors located on the surface. They're also very susceptible to enzymatic degradation. So proteolysis circulating in the plasma or protease in the tissues can degrade peptides, quote, chew them up. So that's why many peptides have rather short plasma half-lives. Uh, Many endogenous peptides, your own peptides, only persist in circulation on the order from minutes to hours. And this also affects your bioavailability. Enzymes can degrade peptides before they're absorbed or before they have the desired effect in the body. So, in all, these properties strongly influence delivery strategies, formulation approaches, and dosing considerations when it comes to peptide regimens. So, let's talk about pharmacodynamics of peptides. Pharmacodynamics describes what a molecule does to the body. Uh, some key pharmacodynamic characteristics of peptides include that their biological effects occur through receptor binding, as I mentioned, triggering intracellular cascades, cellular functional response, and then further signaling out to other cells. What's important to understand here is that once this occurs, the peptide no longer has to be present at the receptor on the cell surface for these cascades to be ongoing and the downstream biological effects. They can literally persist for hours or even days after the peptide is no longer present at the receptor or even present in the plasma. So, keeping this in mind, when we talk about plasma half-lives of peptides, it can differ how that affects peptides in comparison with more traditional drugs. Plasma half-life is a very useful way that we try to establish the biological activity duration of more traditional small molecule drugs, but it doesn't apply so well to peptides. Again, because these downstream effects can keep going much long after the peptide is actually present. So what's important to understand here is that there might not be an objective lab parameter that you can actually pull to measure how long a peptide is actually working. And the effects need to be assessed on the functional response scene as opposed to pulling a lab to see how long and how well it might be working. Let's go further into some of the pharmacologic information that is known about peptides and some of the inherent constraints present. So there's a lot of knowledge gaps still about many peptides. As I mentioned, the plasma half-life might not apply well to many peptides, and then in addition to that, it's often based off animal models and not well understood in humans. Uh, the bioavailability data uh, in various administration routes might not be well understood at all, and the receptor occupancy and binding kinetics. uh, might be poorly quantified. And then the duration of intracellular signaling and how long and intense the effects might be from particular doses might not be well understood. So it's important to keep in mind that when it comes to dosing strategies for many peptides, that they frequently rely on strong mechanistic reasoning rather than fully characterized pharmacologic models that we might have with older, more traditional drugs. So let's go further into some practical applications when it comes to peptide regimens and a specific patient. So these knowledge gaps and these particular characteristics that I mentioned about peptides, um, it means that the dosing strategies and regimens are often heuristic, and this means inferred or based on strong clinical expertise and mechanistic reasoning, just kind of what has been working, rather than a defined pharmacologic model that you might have with older traditional drugs. Again, there's challenges in interpreting biological duration and how that might apply to a specific lab parameter. And then there's uncertain dose response relationships with optimal dosage ranges, with how that might vary from patient to patient, with keeping their differences in physiological states in mind. So the optimal dosage range might not be well known initially for an individual patient. And then there's a lot of model uncertainty on from patient to patient because there are different physiological states on how long and intense the effect might be and how to actually measure that. There might not be an objective lab parameter to pull that you can see how well the peptide is working. So this incomplete data requires a very cautious interpretation when it comes to evaluating peptide dosing strategies and the expected duration and intensity of the responses. Let's go further into individual patient-centered considerations regarding peptide regimens. There's going to be individual variability, as I mentioned, with each patient and their physiological states. This might result in them having different receptor expression levels, meaning actually more or less receptors, or they might be more or less sensitive. There's going to be differences in their signaling cascades and the intensity and duration because they're different physiological states. They're going to have different goals. One might be metabolic system oriented, another endocrine, another recovery system oriented, or some other system based focus objective. There's going to be different comfortability with dosage routes. Many peptides, because of those inherent short biological half-lives and durations, require daily administration. Everyone might not be okay with a daily injection. So it's important to consider if there might be an alternative delivery method available to a peptide to facilitate a patient's adherence and comfort. And then when it comes to response interpretations, uh, again, there might not be a lab value that you can actually pull to objectively measure how well the peptide is working. So the assessment needs to be based on a functional response actually seen by the provider and the patient. So in all, it's important to keep in mind that all these limits in the information we know about peptides and these individual patient variabilities require a very individualized dosing, a very individualized formulation, a very individualized administration, and a very individualized assessment of each patient. So let's talk about some different administration routes that are available to peptides. The first is intramuscular. This is going to give you the fastest absorption and the highest peak concentration due to the high vascularity present in the muscle tissue, but it's also going to give you a very quick clearance of the peptide. Uh, and keeping in mind that so many peptides require daily administration, it's not ideal for that. No one was taking IM shot every day. So then we have subcutaneous administration. This is the most common systemic delivery method for peptides. This is to give you a bit slower administration than intramuscular injections, but it's going to give you a very stable exposure and it's more conducive to daily administration. Then you have intranasal delivery. This is where the peptide could potentially be delivered and absorbed through the nasal mucosa. And this is a very useful potential access point for peptides where the desired effect is on the central nervous system, because that can be a bit of a gateway to that region if it can be absorbed through the nasal mucosa. And then there's oral administration. This can be very limited by gastrointestinal degradation, by the enzymes and harsh environment of the gut, but is achievable with some peptides that might have sufficient stability or specialized dosage formulations that enable this. And it's also very useful with the desired effect is actually in the GI tract and not systemic absorption in the body, but you're actually trying to produce an effect and target the GI tract itself. And then there's sublingual and buccal administration. This can potentially avoid first pass metabolism and some of the enzyme degradation and harsh environment of the gut that might affect the bioavailability of the peptide. But this can be very limited by the peptide size and membrane permeability properties. So the absorption rate here is going to vary peptide to peptide somewhat. So let's talk about how peptides might contrast a bit from more traditional drugs. Peptides differ from traditional drugs somewhat in both their structure and function. Again, peptides show receptor-mediated signaling, frequently resembling your body's own endogenous activity, whereas traditional small molecule drugs can often diffuse easily across cellular membranes, and this gives them a broader exposure and distribution to the body's tissues. This creates an environment where they can potentially interact with multiple targets, and their selectivity here varies greatly depending on the compound and drug class itself. So it's key to understand here is that peptides almost always act through defined receptor-mediated pathways, whereas traditional small molecule drugs can vary widely in their selectivity and distribution. So let's talk about how peptides are being used in modern medicine. Peptides are being used widely across multiple areas of modern medicine. Some heavy areas of clinical interest, as most of us know, include metabolic disease, where they're being used in metabolic function and weight management support. They're being used in endocrinology, where they're being used in hormone signaling and endocrine function support. They're being used in oncology for cell signaling research and diagnostic applications. They're being used in immunology for immune function support and inflammatory modulation. And they're being used in reproductive medicine to support reproductive physiology. It's important to understand that there's many peptide-based drugs are currently approved for clinical use worldwide. So how are peptides evolving for the future? Delivery methods are under a lot of investigation evolving right now. Oral delivery methods are being further researched to open up this route more that's so convenient for patients. And there's longer acting formulations in clinical development, so the peptide doesn't have to be dosed so frequently. And there's further research into optimization of receptor selectivity and a more refined physiological response through structural modifications. And then areas of clinical interest that peptides are further expanded into. Again, metabolic disease, where they're being further research in metabolic function, weight management support, and body composition research. They're being further looked at in neuroendocrine and central nervous system applications, where they might have effects on appetite signaling, your response to stress, and your sleep physiology. They're being looked at in inflammation and tissue repair contexts, where they might modulate immune signaling and also facilitate tissue repair. And they're being researched in cardiometabolic systems, such as the vascular system, the renal system, and other metabolic function systems. So in all, peptide drugs are developing and advancing across multiple clinical areas in both approved and investigational agents, and they're becoming more accessible, longer acting, and more receptor specific. In whole, this is extremely exciting for the future of personalized medicine. Here are the references used in this presentation if you want resources on how to find more information on peptides. Thank you for attending Strive sessions today and learning more about peptides. Please stick around for a live Q&A.